that are in our body, we should prepare a smear. For example, studying of the blood, urine, the cerebrospinal fluid, the lymph, the peritoneal fluid, the precordial fluid, all of them, all of things that are the fluid. Yes, we put one droplet of them on the slide. This is a quadrangular glassine that we use in the lab. In all of the lab, we put the samples on them. We put one droplet on it, and by another lamel, spread this one droplet to all parts. Then do a staining that for the blood, mostly we use HRD, or is there another special staining as Gibbs size staining that used for the blood study. Gibbs staining. Mostly what we have used the hematoxylin also. Stain the blood cells and study them. Under microscope, we can see these cells as this picture.
Give someone.
Give some, but just for your knowledge, it's supposed to use the hematoxylin and eosin. Yes? But this process named as the smear. In the lab, these because, as I said for you in the first semester, for studying other tissues, we cutting them by microscope. But for the studying of the fluid, we prepare a smear that take one droplet of the any fluid, put them on the slide, and then push them by another one slide, and finally spray them. Then under microscope, we can see three types of the cells as erythrocyte or red blood cells that are more than other type of the cells, have pink color under light microscope with H&E staining. The second type are leukocyte. You know, the erythrocyte don't have any nucleus, just are some pink color cells. The second type are leukocyte that have nucleus, that again based on the shape of their nucleus and the number of the lupus of nucleus divided to two types that explain in detail later, and the third one are platelet or thrombocyte that involved in the prevention of the bleeding. The first type of the cells are erythrocyte or red blood cells. These cells are biconcave cells that have into, I see here picture, yes, as these, the middle or center part depressed in mouth side, and the peripheral part have bulging. That by this shape, the surface of the cells is more and more, and they can involve for the more exchanging of the blood gases. That this shape is because of the existence of some the actin filaments that are under the cell membrane and some other proteins that link to the actin and keep the shape of the RBCs. They are pink color under microscope, and in the center, because of these biconcave structures, they are white. Light microscope, they have pink color without any nucleus, that we know that why they don't have any nucleus, because you know nucleus is very important for the cells, and we want to know why these cells are active and have a lots of function, why they don't have any nucleus. In the next topic we know about them. And under the electron microscope, transmission electron microscope, we can see also these biconcave structures on the term nucleus. This is the immature RBCs that have nucleus, and the most part of the cells included the cytoplasm. As the other cells develop polysaccharide chain joined to these proteins, just the glycophorins of the cell membrane. And this polysaccharide chain involved in the blood group. In different person, the type of these carbohydrates are different, and because of this, we have different type of the blood group. Because of this protein is important. The another one is a protein band tree, that this protein finally by some proteins attached to the actin filaments and involved in the maintain of the shape of the cells. The RBC shape maintained by cytoskeletal complex. Cytoskeleton, we explained now in the cell that cytoskeleton included the intermediate filaments, microfilaments, and microtubules. And here, the microfilaments as actin filament are necessary for keeping of the shape of the cell. As you know, the actin filament form a network under the cell membrane of all of the cells. Yes, under the cell membrane they form a network, and here show as this. The actin filament attached to another proteins that the important of them are specterin and ankylin, that they join finally to protein band tree and keep the shape of the cell. Because of it, the cytoskeleton of their RBCs is important because they maintain the shape of the cells. Again, the most important of them are ankylin, specterin, and actin filament, that are lots of another proteins that don't necessary for you, but you can study by yourself.
So we don't need to know that.
Yes, the name, the ankylin, specterin, and actin is important, but this one you know about also the muscle tissue, because also they are attached to the actin filaments. The actin filament, yes, forms a network under the phospholipid bilayer, and these actin filaments join to another proteins as specterin, and finally specterin attached to ankylin, and ankylin attached to their band tree. They form a complex of the proteins. Band tree, ankylin, specterin, and actin. And finally, these proteins that are the complex of the proteins of cytoskeleton maintain the shape of the cell. For example, if the specterin mutant or not translate normal structures finally can form some type of the anemia, the shape of the RBCs exchange. As you see here, and I explained for you, the glycophorin proteins and also protein pantry attach to them some carbohydrate or polysaccharide chain. That these polysaccharide chain have different type that when in the lab do some procedure, they can show different type of the blood group as A, B, and O. That the antigens or the type of the carbohydrate, yes, some person have just, for example, the type for the group A, some type of carbohydrate. Some of them are B group, have another type of the carbohydrate. polysaccharide chain is important for this function, and they attach to the glycophorin and bacteria. Because of it, these two types of proteins are important. And here show that how they in the lab they prepare that. You don't need to memorize it.
Anna, is this gene only present on RBCs or any other cell?
What?
The glycoprotein gene,
Yes.
is it only present on RBCs or do we have it in WBCs and other cells as well?
No, in the RBCs.
So only
For the
RBCs,
blood group, yes, just in the RBCs.
so only RBCs can detect in our blood group, not other cells.
Yes. But the other cell in the other tissue have the carbohydrate chain. But in the blood, it's just the RBCs have this.
Glycophorin.
Question?
Glycophorin.
No, the glycophorin is a protein,
Yes, yes.
and bacteria, yes, are transmembrane protein. That this chain attached to them, they form the glycocalyx that I explained in the first in the cell. Yes? The type of these carbohydrates are different,
Okay.
but the all person have the glycophorin and also the bacteria or other proteins are same in the other person, in the old person.
Yes,
Just
I
the
meant on red blood cells only.
yes. No, the proteins also are in other cells. The
But the
phospholipid, all
carbohydrates.
of the cells have phospholipid bilayers and also the proteins. But just the RBCs have the chain of the polysaccharide chain structure. The red blood cells, as you see in here, and they have red color. The red color is because of one type of proteins as globin. Globin have red color in our body and have two types. One type of them in the blood as hemoglobin, hem means blood, and another one in the muscle tissue as myoglobin. Myo means muscle. That both of them give to tissue the red color. These protein have red color. Because of it, the RBCs have this color. Because of existence of the globin. What is the globin? Or hemoglobin protein. As you see here, included 4 chains. Two alpha chain and 2 beta chain that form a circular structure, not as a chainy structure, that form a tetramer of the hemoglobin, 4 chain. That as you see here, they join to the iron or hem that and we also in the lab measure the hemoglobin. Maybe you see some the report of the lab. I don't know, maybe you see. But if you in the future, when you see them, when we want to study, prepare the smear and counting some cells, RBCs, white BC, platelet, each of them have a normal range. Another measurement that show in the lab report is the hemoglobin. Hemoglobin show to us the amount or percent of the RBCs. Why? Because the hem or iron, hem equal to iron. Iron is a ion that join to the hemoglobin and transfer by the hemoglobin to the cells, to the part that they do its function. That again, this ion is necessary for the transformation of the blood gases. There is one type of anemia that is a hereditary, as named as sickle cell anemia. That, as you see here, the shape of the blood cells is as a sickle cell. When this disease occurs in some disease, as you see, as I said for you, the normal hemoglobin included RNA, and also I think you now about in the cell biology, bring with itself some messenger RNA, yes, some code that translate in the cytoplasm. That finally the amino acid form. The basic part of the amino acid are some bases, as 4 bases. You know them as adenine, guanine, thymine, cytosine. Yes, 4 bases. That is the smallest unit of the amino acid. Then they switch together. A lot of them form, for example, the thymine amino acid, the glutamine amino acid, malic acid. Yes, these are the name of the amino acid. As you see here, in the normal, there we have the adenine and thymine that finally they express, they translate to the glutamine amino acid. In the cell cycle anemia, just one base replaced by them together. Thymine and adenine, they pair together, yes? Then one of them replaced by the uracil. And uracil pair is the guanine or cytosine. Finally, resulted to the translation of another type of the amino acid as malic acid. Because of it, the structures of the hemoglobin change from the circular structures from the chain structures and resulted to the cycle cell anemia. As you see here, just one, the changing just one basis. Then this is a very hard disease. The person suffers from anemia. Their blood have a high viscosity. Because of this high viscosity, they stop in the vessels. And because of the change, because of the shape of the RBCs, they trap in the capillary, the narrow capillary, and finally this person suffered from the anemia, the low concentration of the blood gases, because the RBCs cannot involve in the changing of the blood gases in the high speed. And on the electron microscope and in the blood is clear that the RBCs don't have the spherical shape and also biconcave shape. Maybe in some condition, the number, the shape, the color of the RBCs change because of the, for example, decreasing of the blood gases in the environment. The amount of the iron in our food or in our supplementary things that we eat, finally they result to the, some changes, some problems in the RBC shape or color or other one. One of these pathological condition named as polycythemia. Polycythemia means the increasing number of the RBCs that sometimes maybe occur physiologically. You know, in the mountain or in the heights, yes, the blood, the pressure of the oxygen is the low, yes. Maybe it happens, I don't know, you go to the high mountain. When we go to the mountain, a little we have a dyspnea, yes, and we thinking that we cannot breathe, yes. Why? Because the pressure of the oxygen decreased in the high heights. In this condition, physiologically, our body increases the number of the RBCs that can carry more gases for the breathing or other function. Yes, this is mostly occur physiologically. Maybe occur in the some person that migrate from the one country to another country. Yes, all of us are in the Middle East, and I think we don't do, for example, maybe you come to Iran, don't have any problem. But for example, if some person come from Europe to Iran, because the Europe now is a highest countries, most of them are highs and also highest mountains. When they come to Iran or maybe your country, the Iraq, Lebanon, or the Saudi Arabia, because these countries are flat and don't have lots of mountain, this person maybe around 2 or 3 months have this difficulty. The number of the RBCs increase that can help to the body that coordinate for the condition. This occurs physiologically, but maybe in some person, maybe it's pathological. Polycythemia means the increasing of the number of the RBCs. This condition also may be one of the questions. Spherocytosis, anisocytosis. In the smear of the blood, maybe we find some RBCs with different shape or different size. RBC size is around 7 nanometer. If the size of them is decreased or are more than the 7 nanometer, this condition named as anisocytosis. As you see, this one is very big or this one is smaller, more less than the 77 nanometer. The anisocytosis means the existence of the RBCs with different sizes, maybe more than 7 nanometer or less than 7 nanometer. If we find with different shape, as you see here, this is a kidney shape. This is an oval shape. Yes? Different shape of the RBCs named as the spherocytosis. They don't have the round shape with biconcave in the central. They have the other type of the shapes. When they contain color,
Sorry about spherocytosis.
spherocytosis, the existence of RBCs with different shape. Anisocytosis with different size. If you want to memorize and also memorize the aniso and the size, both of them have Z, yes? Aniso, size, Z. Yes, you memorize this one and then you can memorize the other one. Hypocoron. The RBCs have less color. They are have a pink color, but the color is light pink color, or no, maybe the central part is have more wider compared to normal RBCs. This condition named the hypochromic anemia because of the amount of the hemoglobin. The amount of the hemoglobin increase or decrease of the RBCs. This condition, more color or low color, named as the hypochrom anemia. Chrom means color. Yes, a color change. Yes, with different color, maybe pink color or light pink or no, a dark red.
Just change in color or there's change in the amount?
No, under microscope we can see the change of the color, but this resulted from the changing of the amount of the hemoglobin. And because these are in the lab, mostly you can these are useful for the future when you go to the clinic. Yes, that you want to study the report and explain for the patient that, for example, how the hemoglobin, their RBCs or others. Macrocyte, microcyte. The size of the RBCs more than 9 micrometer is the macrocyte. Macro means big cells. Microcyte, small cells. They are less than, fewer than the 6 spherocyte show the diameter of the RBCs. And finally, when the number of the RBCs decreased, generally named the anemia. Anemia is a word that used for the person that suffered from different conditions that I explained for you. Because all of these conditions finally resulted to the few amount of the hemoglobin and RBCs, and when the number of them decreased, resulted to the increasing of the blood changes, decreasing of the color or others, and the person finally have a dyspnea and or maybe cyanosis in their lip or other part. The anemia is a word that generally used for the decreasing of the number of the red blood cells that have different types. Some of them may be occur because of the deficiency of the iron. Yes, my name. Maybe is because of the deficiency of the amount or size or the color of the RBCs. All of these conditions as hypochrom, microcyte, spherocytosis, anisocytosis, polycythemia, all of them generally located in one disease that named as the anemia, because all of these diseases finally the person have a dyspnea or apnea, they have difficulty in their breathing. Why? Because the amount of the blood gases, oxygen, that is a very, very important gases for the blood function, decreased in there because the RBC is unable to carry out their from the blood to their cells. Maybe because their size is not normal, the amount is not normal, or other things. You ask your question.
So the hypochromic anemia, in that does the hemoglobin increase or does it decrease?
Decreased.
Hypochromic.
The amount of the hemoglobin decrease. Yes, they have the vitamin. This show the more magnification of the spherocytosis, different shape as these or these or this one, and here show the anisocytosis with different size. For example, this one or this one or this one. Yes, the size of them is maybe fewer or more than the 7 micrometers. Sorry, this is the now we cannot see. The other type of the cells in the blood are leukocytes. Have different type. Leukocyte or white blood cells. Because they don't have the red color, yes, the scientists when discovered them, named them as the white color or leukocyte. The leukocyte, based of the type of the cytoplasmic granules and the shape of the nuclei, divided to two types again. Granulocyte have a lots of granules in their cytoplasm. One, polymorphonuclear. Their nucleus have more than 2 pieces or 2 lobule, as you see here. Yes, most of the cells have round nucleus. Yes, most of our body cells have round nucleus. Yes, in the granulocyte, leukocyte, yes, in their nucleus divided to some lobules or some pieces, more than 2 pieces, multilobulated, also named as the polymorphonuclear. They have 3 types as neutrophil, eosinophil, basophil. The second type are agranulocyte or mononuclear, as same as to other cells, have just 1 nucleus, but and also the granule is very few. compared to the granulocyte cells, have two types as lymphocyte, monocyte. The granulocyte, yes, have a high amount of cytoplasmic granules. Most of these granules biochemically are protein. Because of it, they have well-developed RER, Golgi apparatus, or mitochondria. Because you know, these organelles involved in the protein synthesis and have lots of granules in their cytoplasm. That some of them are same in these three types of the cells. They are same. They are also in the neutrophil, basophil, and the eosinophil. But again, each of these three cells have a specific granules. But the agranulocyte don't have any specific granules, and the nuclear also is a round. The granules that are in the granulocyte cells have two types: specific granules and general or eosinophilic granules. Eosinophilic granules included some enzyme, as this enzyme that you can see, lysosome, defensin, or others, that are in the all type of the leukocyte, that finally they can digest or lysis the bacteria or microorganism. Yes, for the lysosome, yes, included lots of enzyme. Myeloperoxidase, yes, they have the peroxidase that involved in the metabolization of the hydrogen peroxide. That this hydrogen peroxide is a poison material for us and also for bacteria. When this material produced for the bacteria can kill them. By defensin, they bind to the bacteria and form some fine balls or balls in the bacterial wall, and finally again killed by these the microorganisms. At first, we know about the granulocyte cells or multinuclear cells. The first one is neutrophil, as you see in this picture. Neutrophil is the more amount of the white species. Most about the type of white species. Question: which of these? Neutrophil, for example, monocyte, lymphocyte, or eosinophil is the neutrophil. They are the first cells that migrate from the blood to connective tissue for defense. For example, when inflammation or infection occurs in some part of the body, the neutrophils are the first cells that migrate to the connective tissue for killing of the bacteria or other things. And usually, in each day, around 1 million of the neutrophils die in the connective tissue without any function. The neutrophils are blood cells, I explained for you, in the connective tissue. That are in the connective tissue some cells that are temporary. They are blood cells that migrate from the blood, but they should do their function in the connective tissue. That one of them is the neutrophil. Its size is around double of the RBCs. In the blood smear, for the distinguish of the size of the cells, we can compare the cells to RBCs. RBCs have 70 micrometer. Memorize this one. This is just 7. 7 is good number. Yes. The granulocyte cells, eosinophil, neutrophil, basophil. These three cells, these three cells, yes, their size is double of the RBCs, around maybe 12 to 15 micrometer. They are twice of the RBCs. They included the primary and secondary granules. That in the neutrophil, the primary, as I said for you, also named as eosinophilic granules. That included they are same in the neutrophil, basophil, and for the DNA, and also maybe they cannot identify the more sex of some died person that we can prepare a smear for them. This is another procedure for the identification of the sex. But one of them is this, and when it's a lifetime, we can use the DNA from the bone for this identification. Neutrophil, as I said for you, are the first of the cells that migrate from the blood to connective tissue. How they migrate? There are a lot of neutrophil in the blood that they are flew and they are wander for itself and they are in the blood. When the infection or inflammation or in the normal condition, they want to go to connective tissue, transmigrate to the connective tissue. You know, most of the capillaries in our body attach together. Epithelial tissue, yes, attach together. They should not degrade from themselves. They should not separate from themselves. If they attach together, the neutrophil don't have any way for passing from them. They should rupture this blood or find for themselves a way that can pass from the blood to connective tissue. For this purpose, there are some proteins on the endothelial cells of the capillary. Endothelial cells are one type of squamous-shaped cells that line the vessels, epithelial tissue. Yes, squamous, simple squamous lining line the different of the vessels as capillary. These cells named as the endothelial cells. There are some proteins in these endothelial cells that their neutrophil have the receptors are in the endothelial cells and the neutrophil have some ligand or some proteins that can join to these receptors. How they join to their receptors? When they want to migrate to their connective tissue. The first one is the selectin. Selectin means the selection. When the inflammation or the infection occurs in some part of our body, some chemical material released from macrophage. These are some cytokines as different type of interleukin. Maybe you hear them. They are inflammatory factors. When this factor released from the macrophage, from connective tissue, selectin expressed in the endothelial cells, and neutrophil have ligand for them. Yes, for the selectin. The fellow neutrophil attached to the wall of the capillary. Attachment occurs. Then they move as the walls. Lots of them attached, just not one. Here shows just one for understanding, but maybe thousand of them attached to these protein as selectin, and they ruling follow up together. After that, there is another receptors on the endothelial cells for integrin. That is integrin finally stop the neutrophil and prevent their movement. When they stop, the tight junction or adhesive junction that are between of the endothelial cells open. Yes, they separate. The cells separate from together and neutrophil can migrate from the blood to connective tissue. This process that have different steps, releasing of the cytokines, expression of the selectin, the neutrophil select and attach to their receptors on endothelial cells. Ruling is a slow movement, and finally they attach to the integrin. By this attachment, they losing intercellular junction as tight junction and in lymphocyte are more because these two types of the cell are more involved in the defense, in the immunity of our system. Let's see what we have here. This is the animation. You can just please see this animation. The cytokines, yes, expose the selectin receptors. They attach to these receptors, and finally the neutrophil join to their ligand, and the ruling occurred. After that, another protein exposed for the integrin that they attach to this and lose the junction between the endothelial cells, and they can pass from the blood to the connective tissue. This is show the diapedesis, different selection. The cytokines, selectin, integrin, and finally passing from the blood.
Why do we need integrin if we have selectin? Is there a difference between
There, I think there in the immunology, you have a project about the immunology. But if you can, if you want to more study your medias, you can search in the internet as interleukin or type of the interleukin. I think in the Wikipedia there are a lot of information about it. There are different types. For example, interleukin 1, 2, 8 that are more important and do the most researcher about them. Any question? Yes, we have a rest. Yes.
The endothelial cells, they're like the walls of the blood capillary.
The endothelial cells are one type of the epithelial cells that line the vessels. I think yes, you are from the first. First, in the epithelial tissue, I explained for you. There is in the table that when I explain about the simple epithelial. You are, I think, new student, yes? Yes, 15 minutes, 9:30. Yes. For the polynuclear cell are basophil. Because it included a lots of purple granules named as basophil. Basophil structure in histology means the cells or structures that have purple color. Why they are as this? Because they have well-developed RER and the Golgi apparatus. Their diameter also as twice of the RBCs. Their cytoplasm contain large purple or black specific granules that they are very large and can obscure the nucleus. We cannot identify the basophil under the microscope because the first one, the amount of them is very, very few, just 1% of the leukocyte. And the second one, we are able to identify the nucleus because these granules are very big and cover the nucleus. They involved in the allergic condition, and the nucleus is bilobed. Have just 2 lobules, not more. I think I pass one of them. Yes. The third type, yes, the basophil have the specific granules and general or eosinophilic granules, that these are as same as to other granulocyte cells. But the specific of granules of them included the histamine, heparin, and some chemotatic factors, that these materials involved in the allergic condition. When some allergen from the food, from environment or others, these cells have receptors for, as I said, I explained for you in the plasma cell, in the connective tissue. The function of the plasma cell and basophil are same. Yes, that they have receptors for these allergen or some allergic materials resulted to some allergic signs in owls, such as rhinorrhea, contraction of the respiratory muscle that resulted to the dyspnea or apnea, the hypotension or decreasing of the blood pressure. Eosinophil. It also is a twice size of the red blood cells. These cells also have bilobular nucleus. Thus, we know that the neutrophil have more than 2 lobules, maybe between the 3 to 4. But the eosinophil and basophil, both of them have 2 lobulated nucleus. Yes, that we can identify the eosinophil or basophil from neutrophil. The first one of the landmark. And the second one, that the more than that I explained for you. Eosinophil, its granules against the basophil that have purple color. In the eosinophil have pink or red color. Why? Because the granule included one type of specific proteins as major basic protein that are just in the eosinophil. And because of these, the granules of the eosinophil have red color. We can identify by these red granules these cells. Under electron microscope, these granules have oval shape. In the center, they are electron dense that show this type of their proteins. The function of the eosinophil, they act as an anti-parasite substance. Some parasites, some worms that are in our body, they attach to this worm or parasite and lysis finally whole of his bark or their own bark. And as this, yes, this is the one type of the parasite. As Helmis is a type that you know about them in the parasitology. You have another credit. These cells, the eosinophil, have a receptor for them. As you see, attach and release their granules as this protein and digest and lysis the bark fiber. This is maybe another question. For example, in which of these cells express major basic protein? Eosinophil, basophil, neutrophil, lymphocyte, or other? It's a specific marker for them. Yes, basophil have purple color granules, but eosinophil have red color. Why? Because in these granules in the eosinophil included this type of these proteins. These are the same type of the granulocyte, leukocyte, or the multinuclear, or multilobulated. Yes, the nucleus have multilobulated. Also, the nucleus of the eosinophil is bilobulated. Mononuclear or agranulocyte, they have a few amount of the granules in the cytoplasm, just the eosinophilic or general granules, and also they have just one nucleus, not lobulated nucleus. Have 2 types, as lymphocyte and monocyte. Lymphocyte mostly have a spherical shape and a spherical nuclei, have different size that divided to a small, medium, and large size. And mostly in the blood, we can see the medium size of it. They have 2 types, as T lymphocyte and B lymphocyte. That B lymphocyte, 3 types. B lymphocyte finally migrate to the connective tissue, differentiate to plasma cell, and involve in the production of the antibody, production of the immunoglobulin. T lymphocyte migrate to the thymus gland and involve in the cellular immunity that you know about more detail in the next semester at inshallah I'll come in the lymphoid tissue and also in the immunology. I think in the 4th or 5th semester. NK cells, NK lymphocyte, recently discovered one. Kidney shaped or bean shaped nucleus. That there in some one part, the little is impressed. Yes, the nucleus have kidney shape. One nucleus, kidney shaped or the bean-shaped nucleus. That in the monocyte and also in the lymphocyte, the most part of the cell occupied by nucleus. They have a few amount of the cytoplasm. As I said, in the connective tissue, monocyte can migrate to the other tissues and differentiate to different type of the phagocyte or different type of the cells in different tissues, as Kupffer cells or osteoclast or others that explain for you in the connective tissue. Monocyte has from diapedesis to connective tissue. They form lots of different type of the macrophage. For example, dendritic cells in lymph node, Langerhans skin, Kupffer liver, osteoclast form. This is, I think, you now one of the questions. This is one of the question. Yes. Actually, and the micromelia in the CNS. The same type of the cells in the blood are platelet, involved in the prevention of the bleeding. They are the smallest one of the blood cell. They have around 2 or 4 micrometer. Mostly they aggregated together, and in the cell they are as a clump, as a group of the cell. You can find, for example, 80 for 10 or more than. They form a group of the platelet. These cells also don't have any nucleus. Just I included the cell membrane and cytoplasm. In the cytoplasm of the platelet, there are some granules and some microtubules. That these tubules maintain the shape of the cells that are under the cell membrane. As other cells, against to other cells, that microfilament, as actin filament, keep the shape of the cells in the platelet microtubules do this function. They form a peripheral of the microtubules that maintain the shape of the platelet as this. In the cytoplasm, the granules mostly are in the middle part, in the center, and microtubules are in the peripheral part. We can see under microscope 2 parts, as hyaluromer is a lighter, granulomer darker and included some granules. These granules, the granulomer part that mostly located in center, included some type of the granules as alpha, delta, or lambda. That the alpha contains some factors, some growth factors, and also the fibrinogen. This fibrinogen is very, very important for the producing of the fibrin. That fibrin is a network of the fibers and finally prevent the bleeding. Delta granules contain some ATP, ATP platelet are in the resting phase. But when they damaged, bleeding because of the expression of some factors and changing of the collagen fibers form the granules from the content of the granules released. That, as I said for you, one of them included the fibrinogen. That is fibrinogen changing to fibrin, the fibrin polymerized and form a network of the fibrin in the clotting part. And then after the blood clot form, the macrophage or the lysosomes that are in the granules lysis this blood clot and remove them. And this is just for your knowledge in the histology. But I now in the next semester, in the circulatory system, explain for you completely in the physiology. Don't need to memorize for this for the histology. Briefly, the blood cells have different types. That here show the normal of them. That don't need to memorize now, but gradually you memorize them because you must know the amount of them for your job in the future. The other topic in the blood is the hematopoiesis. Hematopoiesis generally means the differentiation and production of blood cells. Completely general. How they differentiate? In the inside of the bones, long bone, flat bone, or others, there is a cavity that's named the bone marrow. Yes, bone marrow cavity that included the bone marrow is a material that are inside of this cavity. Based on the content of the bone marrow, we have two types of bone marrow, as red one, yellow one. Red one mostly have red color and included the blood cells. Different step of the blood cells that we know about them in the later slide. Gradually, when the person going to aging, in the aged person, the production of the blood cells decreased. And the stroma of the bone marrow replaced by adipocyte cells, bile adipose cells. And you now, I explained for you in the Thursday that the bile adipose cells have yellowish color in the fresh one because these type of bone marrow have yellow color named as the yellow bone marrow. Red bone marrow, red color, activate in the blood cells production are more in the long bone and have few fat cells that they produce the blood that are more in the neonate, in the child, in the young person, and also in the adolescent. But gradually, in the aged person, with aged, yes, gradually replaced in the long bone by yellow bone marrow, has more fat cells. Yes, this is the difference between them. But the production and the differentiation of blood cells done in the bone marrow. In the bone marrow, there are some stem cells that named as they are the multipotent hematopoietic cells. Hemato means blood, poietic means production. Multipotent stem cells are one type of the cells that can differentiate to different type of the cells. We have for the stem cell have totipotent. This is in the parenthesis for your knowledge. We have the multipotent, pluripotent, and unipotent. Potent means they potentially for differentiation. Unipotent, totipotent is just the zygote that can differentiate to all of the cells. The multipotent can differentiate to with more one type of the cells, maybe 3 to 5, as the hematopoietic stem cell. Pluripotent as mesenchyme stem cell can differentiate just to different type of connective tissue, for example. And unipotent or some type of the stem cell can can just differentiate to one type of the cell. For example, the spermatogonial stem cell in the testes can just differentiate to a spermatozoa, other. These cells are multipotent stem cell that in their differentiation event, they produce lots of blood cells. At first, these multipotent hematopoietic stem cells differentiate to two progenitor cells as myeloid or lymphoid. That the lymphoid one just differentiate to lymphocyte, B lymphocyte and T lymphocyte, and the myeloid one differentiate to other type of the cells. The RBCs, platelet, and different type of the vitamin C except the lymphocyte. Yes? Just thus, the lymphocyte B1, T1, and NK1, yes, derived from the common lymphoid progenitor cells. But the other one, all of these derived from the common myeloid progenitor cells. Here show that there are lots of factors, yes, that in your book explain, but it's not necessary for you and anybody ask from you and also ask from me, because now these are now not all specialized. These are necessary, for example, maybe in the future, inshallah, you want to study the oncology. Yes, you want to specialize oncologists. In that time, you should memorize all of them and you should know all of these. But now, as a general physician for you or for me, that I am a histologist or is anatomist, these are not necessary for me, for you and me. Yes? Yes, don't worry about that. That I explain for, not explain for you in the detail. Because these are used also used in the research. Yes, not in the clinic or other parts. Now we want to know how blood cells developed in the embryonic period and also in the adult person, because you know, because of the existence of this type of the stem cell in our bone marrow, the production and development of the blood cells occur in all of us in whole of our life cycle. Red blood cells or erythrocyte, their progenitor cells named as the peroerythrocyte. That the first of them is the hematopoietic stem cell. Yes, all of the blood cells derived from hematopoietic stem cells that they differentiate to myeloid one. The myeloid one for the production of RBCs differentiate to peroerythroblast. These cells have a very big nucleus, basophil nucleus, and well-developed RER and Golgi apparatus. Why? Because these cells involved in the synthesis of the hemoglobin. Hemoglobin is a type of protein. Protein need to RER, free ribosomes, and Golgi apparatus. All of these are basophil structures. The cytoplasm and the nucleus have basophil color, purple color. Yes? These cells named as basophil because the color of it is purple color. The nucleus and the RER and ribosome that are in the cytoplasm involved in the synthesis of the hemoglobin. They synthesis and store the hemoglobin in the cytoplasm and do more and more, that finally spherical shape. But then the cytoskeleton developed and they have Bankiker shape, and the erythrocyte or RBCs are in the cytoplasm. Yes, we can see them. These events occur in the bone marrow, and just we can see 1% of the reticulocyte, around 1%, not more. If they are more, yes, it may be shown to some the cancer of the blood cells, and more of the RBCs are erythrocyte. Just for the development of the RBCs, you should know the changing of the nuclear and changing of the cytoplasm, as you see here. At first, they included a large euchromatin and active transcriptionally nuclei because they want to synthesis of the hemoglobin. Gradually, the nucleus regressed small, the pattern of euchromatin changes to heterochromatin, and finally they don't have any nucleus. About the cytoplasm, at first they have abundant RER. Gradually they have two double color as basophil, acidophil, and gradually they included at first RER and also hemoglobin. Then they have just the hemoglobin, don't have any RER. For the name of these, yes, you memorize just that in the erythropoiesis is a event or process of the production of RBCs, differentiation of RBCs from myeloid cells to reticulocyte and erythrocyte. But just memorize the name of these. Don't need to memorize all of these cells. Just study them. Granulopoiesis is an event, different process, different step for development of the granulocyte, leukocyte, the neutrophil, eosinophil, basophil. The first step of them are same until the production and synthesis of the eosinophilic granules, because the eosinophilic granules are same in them. Then each of them have the specific granules. The parenchymal cells of the granulocyte cells named myeloblast is a blast that want to start the protein synthesis, don't have any cytoplasmic granules. Then differentiate to next step. At first, synthesis the eosinophilic or general granules. That's because these granules included the proteins and some type of the enzyme that they have the peripheral. Until this step, the neutrophil, eosinophil, basophil are same. In the next step, they synthesis the specific granules. That the specific granules have pink color because mostly included the carbohydrate that join to the proteins. And these granules are specific in neutrophil, basophil, eosinophil. Each of these cells have a specific. For example, eosinophil have major basic protein. Neutrophil have lactoferrin, yes, that bind to the iron. And the basophil have lots of purple granules that included the histamine or heparin. Yes, the specific granules are difference between them. But in this step, we can identify them from together. And finally, all of the cytoplasm filled by these granules, the specific one and also eosinophilic one. Their nucleus at first have round shape, but gradually tubulate Now nucleus, lobulated, and finally they mature to the mature granulocyte. In the neutrophil, have more two lobulated nucleus, 3 or more, and eosinophil and basophil are two lobulated. That's again until this state done in the bone marrow, and we can see just the mature cells. In the basophil maturation, when the nucleus want to be lobulated, maybe this lobule attached together as this. We name them as band cell. If in the blood smear we see these cells is shown to us, that we show notice to the patient, because these band cells show to us the starting of the cancer cell or blood cancer that's named as leukemia. Generally, all of the blood cells tumor named as leukemia. That maybe these cells, around 1% of them, as same as to reticulocyte in RBCs, are normal. But more than of the 1% is the pathological condition. Monocytes are derived from the monoblast. They have a very big round nucleus that occupy all of the cytoplasm. The monocyte and lymphocyte are agranulocyte, white blood cells. It means they don't have the specific granules and have a few amount of the eosinophilic granules. They also at first are basophil. The nuclear shape at first have a oval or round shape. Gradually they transfer to the kidney shaped nucleus. Their cytoplasm at first is basophil because at first involved for production of the a few amount of the granules. The granules is few. Gradually, a little the size of the nucleus regress and small, and in some part of the cytoplasm we can see a few amount of the eosinophilic granules, and then they can migrate and should migrate to connective tissue that differentiate to different type of the macrophage and do their function. Lymphocytes are multinuclear, that also they derive from the hematopoietic stem cell. Then they differentiate two common lymphoid progenitor cells, that is progenitor cells differentiate to B cell, T cell, and NK cell. T cell maturation done in the thymus gland, that you know about them in the next semester. B cells migrate to the connective tissue, differentiate to plasma cell, and NK cell also are in the connective tissue that involved in the killing of the tumoral cells. Platelet maturation in the embryonic, in the bone marrow, the first progenitor cells of these cells named the megakaryoblast. Mega means big. skin. The monoblast derived to the monocyte and finally to wandering macrophage in the other tissues, and the lymphoblast, some of them differentiate in the first step of the differentiation of them done in the bone marrow, that finally differentiate to B lymphocyte and to plasma cell. But the lymphoblast that involved in the production of T lymphocyte migrate to thymus gland and their maturation done in the thymus gland. And finally, the megakaryoblast are big cells. They have lots of cytoplasmic process, that these process separate from the cell and differentiate from very small cells that don't have any nucleus or organelles. Do you have any question? Blood and hematopoiesis in your schedule. We have 2 sessions for today. I don't know you have it or not. That 2 hours is for nervous tissue because nervous tissue have 4 hours. And it's the muscle tissue. That today I finished the nervous tissue. If you agree, because we have tomorrow also class together, I see you tired from me. This is just one suggestion, yes? You can conclude with yourself and inform to me, yes? Today I finished the nervous tissue and remain 2 theoretical subjects for you as muscular tissue and skin for the next time. If you agree, tomorrow I finish the theory and also I don't know about the lab. We can, if you have any class in the afternoon or no?
Yes.
Which
Which
time?
3 p.m.
3 p.m. And also tomorrow?
Maybe it's not today. It's
Maybe we're going to change it to the 1 p.m.
Today? No, change some. You have the 1 p.m. to have with me class. Today, you have class with me until 3 p.m. Yes? That I can finish this is just some suggestion that you can give me your idea too. I finished the nervous tissue and we have one practical class for about the working with microscope and we can see some of the slides. Tomorrow in the morning, I finish the theory topic from the muscular tissue and skin, and then we have another practical because we have 2 practical sessions. We can see some of the slides tomorrow, and I finish all of the theory subject, and in the next time that I come to the Kish Island, please study all of the topic that I can review for you, the theory. The part that you have problem, there are some difficulty maybe for understanding. I think if I finish the theory, it's better because most of the students come because I check your list. I think just 5 to 6 students. That one of the new students come now. Yes, that in the revision because when I finish, they also can use around your writing and also the book and study them. That in the next time that I come, just I explain for you the difficult things, that some things that you have some difficulty for their understanding. I think it's better. But in your schedule, today I finish the nervous tissue, tomorrow we have just practical. And I think we have, because the practical is not difficult, and also I think you enjoy from the practical is better. Because you can see under microscope there some beautiful slides, some beautiful picture under microscope. And we can use in the morning for theory, and then we have practical, and I think we can finish around maybe 2 or 3 p.m. If you agree, yes, now we have a rest until